Charles, why is this cereal so sweet?
Honey.
Where does it come from?
Bees.
Where do they get it?
Plants.
What is it made out of?
Sugar.
How do they make it?
Photosynthesis. I'm Charles, and today we're going to talk about photosynthesis.
In the plant cell, there are small green organelles called chloroplasts. These chloroplasts have two membranes, the inner and outer membrane. Within the inner membrane, there is a fluid called stroma, and stacks of interconnected discs called grana. Each membranous disc in a grana is called a thylakoid. Within the membrane of the thylakoids is the thylakoid space. In the membrane of the thylakoid, there are protein structures called photosystems. Photosystems have several light-harvesting complexes, each of which contain the pigment chlorophyll. The light-harvesting complexes surround the reaction center complex, which contains a special pair of modified chlorophyll molecules and a primary electron acceptor. When the sun's light strikes the light-harvesting complexes, light energy is converted into thermal energy, and the chlorophyll molecules are excited. The thermal energy moves from chlorophyll to chlorophyll until the surge finally reaches a special pair of chlorophyll molecules in the reaction center complex. The special chlorophyll molecules donate an electron to the primary electron acceptor in the reaction center complex, and that electron is transported into the electron transport chain.
That's really cool, but where do all those electrons come from? There are enzymes in the thylakoid that catalyze a reaction called hydrolysis, or the splitting of water molecules. During hydrolysis, 2 electrons are released and move into the reaction center complex of the photosystem, and then into the electron transport chain.
As the electrons move through the electron transport chain, they power the movement of 4 protons through the cytochrome complex, another protein in the membrane of the thylakoid. The electrons move to the second photosystem, ironically named photosystem 1, where they are recharged by the light-harvesting complexes. Then, the electrons are ferried to NADP+ reductase, which uses the 2 electrons to bind a proton to NADP+, forming NADPH, an electron carrier the chloroplast will use later. Meanwhile, in a process called chemiosmosis, the diffusion of highly concentrated protons from the thylakoid space to the stroma powers a protein called Atp Synthase, which adds a phosphate group onto ADP, forming ATP. The products of the light reactions, ATP and NADPH, are necessary for the next stage of photosynthesis, the Calvin cycle.
In summary, protons move into the thylakoid space through the electron transport chain. Then, as protons diffuse back across the membrane, they power ATP synthase, which powers the production of ATP.
The products of the light reactions, ATP and NADPH, are necessary for the next stage of photosynthesis, the Calvin cycle.
In the first step of the Calvin cycle, carbon fixation, an enzyme called ribisco fixes carbon onto ribulose bisphosphate. This forms a short-lived intermediate molecule, which soon breaks, forming 2,3-phosphoglycerate molecules.
Then, the 3-phosphoglycerate undergoes a series of changes involving phosphorylation from ATP and reduction from NADPH, which forms G3P. 1 G3P leaves the cycle, and 5 G3P remain to form 3 more ribulose biphosphates, starting the cycle again. The G3P molecule binds with another G3P molecule.
In the conclusion of photosynthesis, we form 2 G3P molecules, which bind together to form sugar, the stuff that makes our cereal sweet.